UPGMA is an important method which is a distance-based method. It stands for unweighted pair group method using arithmetic averages. So it assumes molecular clock hypothesis. So it assumes the equal evolution between different sequences. So the two sequences with the shortest evolutionary distance between them are assumed to be last derived or last evolved or diverged from some common ancestor. So they are kind of correlated in evolution with one another. So it takes the sequences where we have the shortest distances and put them together at first, and then keeps on adding further sequences, and in the end it can come up with an integrated tree. So here we look into this distance matrix. We have A, B, C, D and E. In fact, we have 5 sequences. For them we have these 10 distances. So let's go with the ones where the distance is really small. So for instance, we can take the distance as 10. We can arrange this 10 into, since we believe in equal evolution, so this distance 10 gets divided between these two on their branches between D and E as 5 and 5. So in fact D is equal to E, which is equal to N, or DE, which is actually the distance between these two divided by 2. So total distance was in the matrix was 10. So it is 10 by 2. We get 5 on both of them. So in this way we started the ones which were present at a minimum distance in the whole matrix, and then we have their branch lengths which we assume, and they are, since we have assumption of molecular clock, so these distances are equal to one another. So half of the distance goes in one branch, and that is we represent over here with a small letter D, and we represent with, so in UPGMA we take the closely related pair where we had the shortest distance in the whole matrix. So we take, for example, D and E, they were separated with a distance of 10. So we divide it into equal halves and give it to both of them their branches. So we, for example, we can come up with their tree. We have D and E. The branch which is linked with D is small d, and here we have small e. So we believe that both of them are equal, and the distance, the branch length is given by NDE by 2. So the distance between these two in the matrix was 10. So both of them, they will get 5 each. Same way in the next step, we bring the third one. So we combine D and E with one another in that matrix, and then we take the distance of D and E with the one which is closest to them. So in this case, that closest one is actually the one with C. So the small letter c represents its distance, and we can come up with the distance of DE with C, which is given by n of CE plus n of DC, which is the distance between C and E plus the distance between D and C, and we divide it by 2. So in this case it becomes 18 plus 20 divided by 2. So they are at a distance of 19. Since we assume that these distances should be cumulative, so we split that distances. So from D to this common ancestor, we have the distance that 9.5, and this 9.5 then is added to the distance which was here 4.5, and we know that here the distance was 5. So in this way total we should have a distance of 19 towards both of them, whether we go this way or whether we go this way. Same way we can add up the next one, which was B, so the distance, and same way we can take A and B together and we put them together. So as a whole we keep on solving the puzzle, and finally we reach to a tree where we can see that those distances between any two nodes or those any two leaves is sum of the distances on those individual branches. And we have put all of the ones which were close to one another, and then we keep on adding the distant ones, and in this way we came up with the final tree. So UPGMA assumes constant mutation rates, molecular clock hypothesis. Here we take the sequences which are at the shortest distance with one another, we arrange them first, and we make their tree, and we keep on adding then a further distance species onto this tree.